We first see the normal mode of neuronal impulse conduction, which proceeds along the nerve axon from a stimulated nerve cell and bounds from one node of Ranvier to the next. The CNS contains small venules which get leaky in response to a variety of different factors, including activated T cells, which recognize antigens expressed in the CNS, enter the brain, and initiate the inflammatory lesion. This disturbs the blood-brain barrier and allows lymphocytes to squeeze out of the leaky venules into CNS tissue where they proliferate. Then the lymphocytes and plasma cells produce immunoglobulins, which start to break down the myelin sheaths of the nerve cells. This produces changes in electrical conduction along the neuron, since the insulating myelin is broken down. This leads to many symptoms. Chronic lesions develop after a series of repeated inflammatory acute attacks, leading to whole segments of axons being totally demyelinated. This is accompanied by a proliferation of astrocytes in the damaged regions to form gray or pinkish plaques. Repetitive waves of acute attacks lead to stripping away of the myelin, damage to oligodendrocytes that normally help produce new myelin, and a buildup of astrocytes into plaques, forming a chronic lesion. This disturbs or delays nerve impulse conduction and results in impaired motor and sensory functioning.